So here, several intermediate compounds which are produced in Krebs cycle. Usually Krebs cycle is anabolic pathway. It involves the breakdown of pyruvic acid into carbon dioxide and water. And intermediate compounds which are produced, they are in turn used in the synthesis of organic compounds. See, when these are used as respiratory substrate, these are broken down and energy is released, lot of energy is released. Now, in a cell, okay, if fats are to be synthesized, in a cell fats are to be synthesized, you know, this fatty acid is converted into acetyl coenzyme. Glycerol is converted into 3-phosphoglycerol. Interrelationship among the different metabolic pathways showing respiratory mediated breakdown of organic substance into carbon dioxide and water. During that I have discussed. Okay, suppose if the cell is in need of fats, cell has to synthesize fats from external source not there. In that case, what happens is this acetyl coenzyme is withdrawn. It is withdrawn from respiratory pathway. It is withdrawn from respiratory pathway and it is used in the synthesis of fats. Earlier, what happened during respiration? Fats were broken down into fatty acid and glycerol. Okay, this will take part in respiration. That is Krebs cycle all that. Suppose if cell is in need of fats now, the external source of fat is not there. Then what is there? Acetyl coenzyme is there. This has been produced from this after the breakdown of fats. So this acetyl coenzyme, which is taking part in respiratory pathway, is withdrawn from the respiratory pathway, and in turn, this acetyl coenzyme can be used in the synthesis of fats. So initially, it can involve breakdown of the complex organic molecule to form simple molecule. But later on, when the cell is in need of what we call that fat or protein, so such simple compounds can be used, and in turn, that result in the synthesis of either protein or fats. So fats can be synthesized from acetyl coenzyme, okay, and proteins can be synthesized from amino acids. So it involves both catabolism, that is breakdown of the complex molecule, and also it involves synthesis of molecules, that is anabolism. That's why this respiratory pathway is known as amphibolic pathway. Okay, few examples I will give. The intermediate compounds which are produced during Krebs cycle are in turn used in the synthesis of several other organic molecules. Acetyl coenzyme. This is produced from preparatory step. Pyruvic acid is converted into acetyl coenzyme. It is a 2-carbon compound. Okay. This acetyl coenzyme act as a raw material for the synthesis of. It acts as a raw material for the synthesis of carotenoids, terpenes, and a plant hormone, that is gibberellins. So this is one example. The intermediate compound which is produced during Krebs cycle can be in turn used in the synthesis of other organic compounds. So this is anabolism. Then second one, succinyl coenzyme. During Krebs cycle, this product is formed, succinyl coenzyme. Right? This succinyl coenzyme is used as a raw material. It is used as a raw material for the synthesis of what you call chlorophyll, this is pigment, chlorophyll, and then you have cytochromes. Cytochrome electron transport system, cytochrome B, cytochrome C1, cytochrome A, cytochrome A3. Different types of these cytochrome we have come across, which are situated on the inner membrane of the mitochondria on crystal, which act as electron carriers. Okay, they are iron-containing pigment protein acting as electron carrier. Chlorophyll basically present in chloroplast involving photosynthesis. So these organic compounds, okay, pigments, can be synthesized by using the raw material, that is succinyl coenzyme. Succinyl coenzyme is produced during what you call Krebs cycle. Alpha ketoglutarate acid to succinyl coenzyme. That is 6-step. There it is produced. Then another example I'll give. Third one, that is oxaloacetic acid. This oxaloacetic acid is going to condense with acetyl coenzyme to form first stable 6-carbon compound citric acid. Starting of the Krebs cycle, first step itself, right? So this oxaloacetic acid is the raw material. It is the raw material for the synthesis of alkaloids. Alkaloids are organic compounds produced in plants. Okay. Simple example, you have what we call reserpine, serpentine, okay, vincristine, vinblastin. So many different examples you can give. Okay, alkaloids, and you have what you call pyrimidines. Alkaloids and pyrimidines, you have nitrogen bases, pyrimidines and purines. They are present in nucleic acid, DNA, RNA. Okay, so synthesis of alkaloids and pyrimidine, nitrogen bases. These are nitrogen bases. Okay, pyrimidines. Right, pyrimidines, cytosine, thymine in DNA, cytosine and uracil in RNA. So such nitrogen bases can be synthesized. Even alkaloids can be synthesized using oxaloacetic acid. That is one of the key intermediate compound taking part in Krebs cycle, right? So one more example we can have here, that is alpha ketoglutarate acid. Alpha ketoglutarate acid. So when you take this alpha ketoglutarate acid, it is the raw material for the synthesis of amino acid. Different amino acids are produced by using this alpha ketoglutarate acid. Okay, a process called what you call reductive amination. By using that process, using this alpha ketoglutarate acid, glycine can be produced. Glycine is one of the important amino acid from which 17 other amino acids can be produced by reductive amination process. Okay, so alpha ketoglutarate acid is the raw material for synthesis of amino acids. So the various intermediate compounds produced during Krebs cycle are either used in the synthesis of several other organic compounds. That's why it is anabolism. It not only involves the breakdown of complex organic molecule, but also involves the synthesis of other organic compound. Hence, respiratory pathway, respiration, it involves both catabolism as well as anabolism. That's why it is known as amphibolic pathway. So try to remember. So particularly Krebs cycle, we call it as amphibolic pathway because these are all the products produced in Krebs cycle. Okay, so this is about amphibolic pathway. Now last concept of this chapter, that is the respiratory quotient. So very important concept now. Respiratory quotient, it is also known as respiratory ratio. Respiratory quotient, or it is also known as respiratory ratio. So what is this respiratory ratio? See, it is the ratio. It is the ratio of what? Respiratory quotient is the ratio of carbon dioxide evolved or liberated to the oxygen used, utilized, consumed, whatever it is, used, consumed, utilized during respiration in a cell or in a tissue, whatever it is. So the ratio of carbon dioxide evolved, carbon dioxide released, carbon dioxide liberated to the oxygen used or consumed or utilized during respiration in a cell or tissue in what we call living organisms, some living plants, that we call it as respiratory quotient or respiratory ratio. So how we will write respiratory quotient, or Q is equal to volume of carbon dioxide evolved, liberated. Respiratory product is carbon dioxide, is it or not? Carbon dioxide and water. So ratio of volume of carbon dioxide evolved. So you can use a volume of carbon dioxide evolved to the volume of what you call oxygen utilized or used or consumed, volume of oxygen what you call consumed, used, utilized, whatever, right? So this we have to remember. This is the respiratory quotient. It is also called respiratory ratio. So it is the ratio between the volume of the carbon dioxide released to the volume of oxygen consumed used during respiration in a cell. That is called as respiratory quotient. Okay. So RQ value varies. RQ value varies from one substrate or one kind of substrate to other substrate other substrate. So RQ value varies from one kind of substrate to other substrate. See, it means it varies. RQ value of carbohydrates is different, protein is different, fats is different, organic acid is different. RQ value in aerobic respiration is different. RQ value in anaerobic respiration is different. So different RQ values we have to remember here. RQ of carbohydrates. RQ of carbohydrates, we say RQ is equal to 1. Remember, RQ value of carbohydrate is equal to 1. The right reaction. So first we'll discuss all the aerobic conditions. So here C6H12O6, 6 oxygen, product side 6 carbon dioxide plus 6 water plus energy. This is aerobic respiration reaction when what is this? Glucose, carbohydrate. The carbohydrate used as a respiratory substrate. Use this RQ of carbohydrate. Using this, we can calculate volume of carbon dioxide evolved, released, or liberated, whatever it is. So RQ is equal to volume of carbon dioxide released. What is the volume of carbon dioxide released? 6. What is the volume of oxygen used? 6. So RQ is equal to. So RQ value is 1 because volume of carbon dioxide liberated is equal to volume of oxygen used. That's why RQ value is 1. Either it is normal carbohydrate or sometimes see carbohydrate means we have rice, jawar, okay, rice or paddy, jawar, wheat, maize. So usually these are all cereals. These are cereals, barley, oat, etc. They are all cereals. Cereals are rich in carbohydrates. Either you take normal cereals or sprouting. Sprouting means germinating cereals. They are producing small seedlings. Okay, either it is sprouting or germinating what you call these cereals or normal cereals. Normal. They are rich in carbohydrate. So RQ value, either they are normal seeds, normal what you call the seeds of cereals or sprouting or germinating cereals, RQ value will remain 1. Don't get confused. So to confuse you, they will ask, what is the RQ value of sprouting or germinating barley? Barley is cereal. Cereals are rich in carbohydrate. So carbohydrate RQ value will be 1. Now, next RQ value of fats. RQ of fats, lipid. We say RQ is equal to 0.7. RQ is equal to 0.7. So, for example, tripalmitin fat. Tripalmitin fat. Okay, so what is this RQ value of tripalmitin fat? This is the reaction. This is tripalmitin fat. Plant source have this tripalmitin fat. Okay. So we have used a big number to calculate this. So this is how we can balance. RQ is equal to volume of carbon dioxide liberated. What is the volume of carbon dioxide liberated? 100. volume of oxygen consumed, how much? 1.1. You check the balance or not, you have to check now. RQ is equal to 0.7. Equation is balanced or not, check. Carbon 102, then hydrogen is like 98 into 2, 196. 98 into 2, same. Oxygen is check. Now you see here. Why RQ value? See, usually how we write RQ of fan? Usually what we say, RQ is equal to less than 1. In this way we write. This is the exact value. Usually what we write? Less than 1. RQ is equal to less than 1 we will write. Less than 1 means how much there? 0.7. So why the RQ value is less than 1 here, or 0.7? See here, this is substrate, respiratory substrate. Okay, this respiratory substrate is a very long hydrocarbon chain. How long? Carbon skeleton, 51 carbons are there. 51 carbon. Like this, you have written 51, the long chain hydrocarbon. Then you take oxygen. It is having very long chain carbon, but poor in oxygen. Very poor in oxygen. When substrate is poor in oxygen, it needs more oxygen for its breakdown. That you have to keep it in mind. This substrate is a long chain hydrocarbon having C and H more compared to oxygen. Oxygen only 6. Substrate is poor in oxygen. That's why for its breakdown, it needs more oxygen. See here, carbon dioxide evolved is less than the oxygen used. Oxygen used is more because this substrate is poor in oxygen. That's why it makes use of more oxygen for its breakdown. Hence, RQ value will be less than 1. Remember this, very important. Why RQ value of fat is less than 1? Because substrate is poor in oxygen. For its complete breakdown, it needs more number of oxygen. That's why RQ value will be less than 1. Similarly, RQ of proteins. RQ value, respiratory quotient, respiratory ratio of proteins. We write usually RQ is equal to less than 1. Remember, protein also less than 1. In protein also, carbon, hydrogen length will be more. Carbon skeleton will be more compared to the number of oxygen present in the protein. Protein is also poor in oxygen. That's why RQ value of protein is less than 1. So RQ of protein is 0.9. Remember, 0.9. RQ of protein is 0.9, less than 1, 0.9. So whenever substrate is poor in oxygen, then they need more oxygen for their breakdown. That's why RQ value will be less than 1. That remember. Now, so only this given in NCERT. Few more we will discuss. They have given only sugar, carbohydrate, protein, and fats. They have given. Now next, RQ on organic acids. RQ of organic acids. So usually what I will write, RQ is equal to greater than 1. RQ value is greater than 1, more than 1 in case of organic acids. Say for example, balanced or not. This is malic acid. Balanced equation. So when organic acid like malic acid is used, malic acid is used as a respiratory substrate if RQ is greater than 1, that is 1.33. Remember. Another example we can have. Check balanced or not. Balanced equation. Balanced. So this is oxalic acid. You can write in this also C2 H2 O4. C2H2O4. This is oxalic acid. Okay. So RQ value of oxalic acid is 4. More than 1 at all. So it is 4. Malic acid 1.33. Oxalic acid is 4. Remember. Now, why RQ value is more than 1 in case of organic acid means that substrate you see properly. Substrate is having few carbon, more oxygen. Cr, C4O5, C2O4. So these organic acids are rich in oxygen. When the substrate itself is rich in oxygen, for their breakdown, they need less oxygen. They need less oxygen. What you are sleeping on? Huh? So when the substrate is rich in oxygen, organic acids are very rich in oxygen. That's why they need less oxygen for their breakdown. Hence, RQ value is greater than 1. Then RQ in succulent xerophytes. RQ value in succulent xerophytes. Succulent xerophytes, cactus, opuntia, etc. Bryophyllum, all that. Okay. See, in these succulent xerophytes, stomata is open during night time. Is open during the night time. Okay. So during night time, what happens? The carbohydrate is broken down into a organic acid without liberating carbon dioxide. So sugar is broken down during night time when stomata is open into the organic acid. So with organic acid it is malic acid. And there is no liberation of carbon dioxide. That carbon dioxide is used for synthesis of carbohydrates. When the substrate is broken down, okay, that is not releasing carbon dioxide, it is releasing organic acid, that is malic acid. Okay, not balance. Balance. So oxidation of the respiratory substrate during night time when the stomata is open, resulting in the formation of organic acid, not liberating carbon dioxide. Carbon dioxide is not released. So what we will write? RQ is equal to carbon dioxide released 0. Oxygen used. So RQ is equal to 0. So remember, RQ is equal to 0 in case of succulent 0 points. Carbon dioxide is not released. Okay, carbon dioxide is used for photosynthesis, that not released. Okay, there organic acid is produced. No liberation of carbon dioxide, that's why RQ value is 0. Remember. So these are all under which conditions? Aerobic conditions. These are all under aerobic conditions. Now, RQ in anaerobic respiration. We will write that alcoholic fermentation. This is alcoholic fermentation. Energy only 2 ATP molecules produced here. Okay? See that now. RQ is equal to volume of carbon dioxide liberated, volume of oxygen used, 0. Chaitra. Hey Chaitra, why so late? What is the RQ value now? Not defined means? Infinity. So RQ value is infinite or infinity. RQ is infinity in so remember this. Okay. See, in a living system, in a living cell, usually mixed respiratory substrate with them. Mixed respiratory substrate. Respiratory substrate is subjected to oxidation. Usually in living cell, pure protein, pure fats are not used as respiratory substrate. Because in a cell, only carbohydrate will not be there. Protein will also be there, fat also will be there. So in a living cell or in a living system, usually mixed respiratory substrate will be there. Pure protein, pure fat alone are not used as respiratory substrate. You have to keep it in mind. Pure protein or pure fats are not used as respiratory substrate. Usually in a living system, mixed respiratory substrate will be there. So when it is mixed respiratory substrate, it can vary from 0.8 to 0.9. Mixed means cell has carbohydrate, protein, fat. Mixed. Pure substrate will not be there. Usually. Okay. So mixed will be there. So when it is mixed, then RQ value varies from 0.8 to 0.9. Right? This respiratory quotient is measured. It is measured by using an instrument, a device. Okay. So which is that device? Anybody can name it? in the next class, I want to tell me that RQ value is measured by using a device or instrument. Okay, so name that instrument. In the next class, I want to tell me. So this is all about respiration topic. Okay. One difference they will write that is not there in NCERT. Difference is oxidative phosphorylation and photophosphorylation. Oxidative phosphorylation, photophosphorylation. Difference will write. This oxidative phosphorylation it occurs in on higher organisms. Okay, eukaryotic whichever are there here. Usually it occurs in all higher organisms, plant, animal, whatever it is. Here it occurs in plants, in green plants, photophosphorylation. It occurs in green plants. It occurs in what you call crystal or mitochondria. Mitochondria, crystal which has a not F1 particle, no, there. Here it occurs in inocoid membrane or chloroplast. It occurs in thylakoid membrane or chloroplast. Here, oxygen is used. Oxygen is used. Okay. Whereas here, oxygen is liberated, not used. It is liberated. Yeah. Splitting of water molecule will be there during photophosphorylation. Synthesis of ATP using synthesis of ATP using energy of food, breakdown of food, energy breakdown of food, food breakdown, organic molecules will be broken down. That energy is used for synthesis of ATP. Here, synthesis of ATP usually using which energy? Light energy. Synthesis of ATP using light energy. So there, radiant energy is used in the synthesis. Here, organic molecule which are broken down, that energy which is released, that can be used. Right? ATP released is used for metabolic activities. ATP released, ATP liberated is used for various metabolic activities. Okay, so here ATP released is used in their lab reactions. They are used in dark reactions. Fixation of carbon dioxide, Calvin cycle, sugar synthesis. Their ATP can be used. Is it or not? Okay. Then any other difference? Anybody? Any other difference? Here, electron transport. Electron transport, which is there, no? That electron transport occurs in simple manner, simple way. Here, electron transport, cyclic, non-cyclic. Electron transport in inner membrane of mitochondria. Okay, electron transport occur in inner membrane of mitochondria. Here, electron transport occur on thylakoid membrane. Is it or not? Electron transport occur on thylakoid membrane in a cyclic or non-cyclic way or pathway. Here it does not involve cyclic, non-cyclic. Okay, electron transfer to occur. NADH2 or FADH2 on oxidation, electron will be transported finally to oxygen. Right? So electron transport occur in inner membrane of mitochondria. So this we usually call it a terminal oxidation. Okay, usually we call it a terminal oxidation. Here it occurs by either cyclic or non-cyclic way of pathway. Right? So these are few differences you try to remember between this occur oxidative phosphorylation. It occurs during respiration. Is it or not? It occurs during respiration. This one it occurs during photosynthesis. Photosynthesis occurs during photosynthesis. Okay, so these are some of the what you call difference between oxidative phosphorylation and photophosphorylation. So with this chapter is over. Respiration in plant we have completed. Now last chapter of your syllabus. What is that? Plant growth and development. In that plant growth regulator we have to learn. Remaining part all related. Growth, development, photoperiodism, vernalization, seed dormancy, these parts are deleted for theory. last chapter, plant growth and development. So in this, we have growth. Growth is indeterminate in plant. Growth is measurable. Phases of growth rate. Okay. So all these parts deleted. So complete growth and development. This part is deleted for theory syllabus. Okay. Other parts which are deleted here, photoperiodism, vernalization, seed dormancy. So all these parts are deleted. So only one part is there. You are checking whether I am telling correct there. Next, what is there then? Plant growth regulators. This is there only for our syllabus. Hey, delete everything will be there. Afterwards, we will teach deleted syllabus also. First, let us complete what is there for theory. Afterwards, need point of view we will discuss. You need not worry. So we have to start now. Plant growth regulators. These are also known as phytohormones. These are also known as plant hormones. Different names are given to them. So here we have plant growth regulators. Now let's see. These plant growth regulators, simple, diverse, simple molecules, simple organic substances with diverse composition. They are simple organic molecules with diverse chemical composition. They are simple organic molecules with diverse chemical composition. Okay. See, what are these plant growth regulators or phytohormones or plant hormones? They are organic chemical messengers. They act like organic chemical messengers. Okay. So these plant growth regulators are small, simple organic molecules with diverse chemical composition. Okay. So they are organic chemical messengers produced in plants in one part or one region and transported to the other region. They are organic chemical messengers produced in plants in one part or region and transported to other region where they show their physiological effect. Physiological effects. They are organic chemical messengers. They are organic substances produced in one part of the plant, translocated or transported to other part of the plant, where they show their effect, physiological effect. How in human being, we have endocrine system. Gland will be there in one part. Okay, that secrete hormone. That hormone is transported through the bloodstream. It reaches the target organ where it shows its effect. Like that, these are organic substances which are produced in one part. Say shoot tip, root tip, leaf tip, flower, fruit, seed. In different parts they can be produced, and they are transported to other parts of the plant. There they can show the effect. So this is simple definition of what you call the plant hormone or phytohormones, growth regulators. They are of diverse chemical composition we have written. Okay, so they are different what we call derivatives. They are derived from different sources. They are derived from different sources. So one is indole derivative. Indole derivative. Indole derivatives are mainly what we call auxins. Auxins. Okay, auxins are plant growth regulators. Okay, I will write the names. These plant hormones, five, I will name auxins, gibberellins, cytokinin, ethylene, abscisic acid. These five are there, right? So auxins are indole derivatives. So if you have indole 3-acetic acid, IAA, it is one of the naturally occurring oxygen. Indole 3-acetic acid. Indole acetic acid. In simple way, you write indole 3-acetic acid or just indole acetic acid. Indole derivative oxygens. Indole compounds, oxygens. Okay. Then we have adenine derivatives. Adenine. Adenine, it is a nitrogen base. It is purine. It is purine. Adenine derivatives. Okay. So here we have 6. Okay, N6 we can write N6. N6 furfuryl aminopurine. So it is cytokinin, cytokinin or kininin, whatever that. Adenine derivatives are cytokinin, kininin, chemically N6 furfuryl aminopurine. Then adenine derivative, adenine nitrogen base, double ringed purine, from which the cytokinins can be derived. Okay, chemically that is N6 furfuryl aminopurine. These hormones are produced in very small concentration. They are very effective in small concentration itself, in molar concentration. Okay, 1,000th of molar concentration, 10,000th of molar concentration. In extreme small concentration itself, they are effective and they show different physiological effects. Okay, so this is adenine derivative. The next one we have carotenoid derivative. Carotenoid derivative. It is abscisic acid. Abscisic acid, that is carotenoid derivative. It is derived from carotenoid. Okay. Then next we have terpene derivative. Terpene derivative. Terpene derivative we have mainly gibberellins. Gibberellins. Okay. The only gaseous plant hormone, only the gaseous plant hormone, that is ethylene. Ethylene is the only gaseous plant hormone. So remember these different derivatives, right? So these are five types of plant hormones. These plant hormones, broadly based on their function, we group as one growth promoter and another one growth inhibitor. Right? So this will continue on Tuesday. I'll continue. So I need around 3.5 hours to complete these hormones. Maximum 4 hours. Right?